Welcome to Progenesis Academy webinar session 53. For those who are not familiar with Progenesis Academy, we are an educational program focused on embryology and reproductive genetics. Our goal is to foster collaboration and education of the IVF community. If you have attended our webinars, make sure to claim your certificate of attendance. Most of our webinars are honored by AAB for CU credits. If you have missed some of these webinars, you can see them all on YouTube channel or on our website, Progenesis/Academy. Today's topic is the prospect of genome editing in reproductive medicine. We will have 3 oral sessions followed by a roundtable discussion. Introducing Dr. Albertini, Editor-in-Chief of JARC, Journal of Assisted Reproduction and Genetics. Dr. Javed, he's the lab director at Original Clinic and Women Health Center. Dr. Stadmauer, reproductive endocrinologist at Fertility Care, the IVF Center. And Dr. Sharara, he's the medical director at Virginia Center for Reproductive Medicine. And today we're going to be talking about CRISPR technology. So we're going to be defining what CRISPR is. Then we're going to talk about the use of CRISPR in human embryos, the first trial that was done in China. Then we're going to talk about the potential of CRISPR in IVF that include repairing mutations or mitochondrial mutation and other application in IVF. Then we're going to talk about the ethical consideration when it comes to CRISPR technology. So CRISPR first discovery was in 1987. It was discovered by researchers at Osaka University. And what they found is they found in bacterial DNA repetitive regions. Those regions are called Clauser regularly interspaced short palindrome repeat. It's a long title, but really what it means is repetitive regions in the genome. But these regions were intercepts. They were separated by unique sequences. And back then, they did not know what these sequences are. Later on, researchers in the field tried to sequence those regions, those interspaced regions, and they mapped them to known genomes. They found that those sequences map with viruses or phages. Really, the second jump in the field of CRISPR technology was with these 2 Nobel Prize scientists, Jennifer Doudna and Emmanuel Charpentier. And what they've done is tremendous. Basically, they revealed the mechanism of CRISPR technology. Basically, what you see here is bacterial genome. You see some tandem repeat at the bottom, and the spacers are the sequences that are specific to certain viruses. And then on the left-hand side, you see Cas protein that is designed to recognize a specific site in the genome and cut it. And when you cut it, you can either insert a base or remove a base to introduce a genetic modification. There are versions of Cas protein, depends on what bacteria you're talking about. Sometimes you're able to harvest bacteria from the wild, and then you can sequence it without knowing what that bacteria is. You're probably not able to even culture it. And by doing that, they've discovered a variety of Cas protein that have the same role, but they have different configurations. Some of them are multiple genes, and some of them are only one gene. And so it was till 2018 when CRISPR technology was put in practice in human IVF cases. And this was done by a scientist in China, Jiankui He. What he did is basically introduce CRISPR enzymatic cocktail during fertilization. The purpose of this study was to help the future children be immune to certain mutation, and that mutation is a CCR532, which is a receptor for HIV virus. the receptor will no longer be able to be used by HIV virus. On top, the unmodified sequence of that receptor shows a long sequence with the 3 bases, and then the delta 32, the next one at the bottom, is a natural mutation that confer the resistance to this virus, to HIV virus. And it's a deletion of a 32 base pair. What these scientists did was creating a new mutation that didn't exist in human population, which is a deletion of 15 base pair. And he did another configuration, which is a deletion of 4 base and 1 base. I am really surprised that he did not try to replicate the natural variants that is resistance to this virus that it already exists in human, and he decided to create something that is very new. This is the clinical trial coming from that study withdrawn for reason of lack of safety and validity. And just to summarize quickly, the challenges of CRISPR technology is off targets, meaning that you are aiming to change one specific site, but because the sequences may have some similarity with other regions of the genome, you may be modifying in other places, and that's what we called off targets. Another very important aspect is safety. You know, the safety aspect is really an important one, because when you do this kind of modification in genomes, you have to be able to test it in animals and show that it does not affect the quality of life of the individual that has been genetically modified. I am going to wrap up here and then move to our next speaker, Dr. Javed.
Thank you, Nabil. Well, it is my pleasure again to speak with you today, and I am happy to be with so knowledgeable participants. I am just embryologist, so my background, my knowledge of genetics is reasonable, but not anyway close to expertise. So I will be talking on the general aspects. And could I have the first slide, please?
Yes.
Okay, I wanted to just explore what is the history of DNA, and then I would move on to what the status of this technology or research is today. Nabil's talk was very, very, I should say, deeply technical, and for embryologists, we really need to look into what will happen to the embryos or to our generation. So if we look at the history of the DNA, so it was first isolated in 1869. And then in 1944, it was known that this DNA is responsible for heredity. In 1953, its structure was discovered by Watson and Crick. I think everybody knows those are the Nobel laureates. In 1970, enzymes were discovered to cut the DNA, to edit the DNA. And in 1975, the DNA sequencing was developed. This, I think many of the embryologists will know that Sanger sequencing is very, very popular. In 1990, for the first time, PGD was used to check the embryos. In 1993, this new editing technology was first talked about, and in 2001, this technology was given the name CRISPR, which is very popular and under a lot of discussion and debates these days. The human genome draft was published in 2000, and in 2003, human genome was completed. In 2012, cell DNA was edited by CRISPR, as Nabil said. In 2018, the human genome was edited by this scientist, John Kui He. Can I have the next slide, please? So this slide talks about how we have used this technology for the clinical use. As I said before, in 1990, the PGD was used for detection of sex-linked diseases. In 1992, the single gene disorders were detected by this. In 1997, there was birth from mitochondrial transfer. So this we call it three-parent baby. Some people call it designer baby. In 2000, a baby was purposely chosen to be HLA match and free from Fanconi anemia. In 2016, live birth happened after the maternal spindle transfer. In 2017, live birth happened after the pronuclear transfer. And in 2018, the human genome was edited by Dr. He. And of course, after this, he was punished and he was prisoned. Next slide, please. So what are different kinds of genome editing? So we can edit the human genome at different levels. The first level is the somatic cell genome editing, like the body cells can be edited. And this, to me, would be more publicly accepted because this is affecting only that person who has defective genes. And this has already been used for treatment of sickle cell anemia and any other, some of the other single faulty genes. These kind of changes do not pass to the future generations. Only the treated individual will be corrected. But unfortunately, at this time, the cost of this treatment is very, very expensive. It is about 1 million per patient. The second, which is very kind of sensitive area, is the germline editing. So in this editing, the eggs, sperm, their precursors, and embryos can be edited. But these changes made in these cells will pass on to the future generations. At this time, this gene editing is inhibited almost all over the world. The reason being our knowledge is still not complete, and we do not know, as Nabil said, off-target changes in the DNA. So I think at this time, the scientists agree that the experiments should be conducted on the laboratory animals. And once we know what we are going to do and what will be the consequences, then at that time, we can probably move on to the human. The third kind is human epigenetic editing. In this way, the DNA is not changed, but the activity of DNA is changed. Next slide, please. Why we do, or why we like to do the gene editing? The first thing, of course, would be medical reasons. And if there is a compelling medical reason, the science is there to explore it and to find a cure for this. However, in such cases, the risks have to be addressed thoroughly. And also we should see that if there is another easy alternative and where there is no other higher risk, then if there are other alternatives, then we should not really edit the DNA. And then we should follow up for a long, long time after we have edited the DNA to see what are the consequences. And it should be really strictly controlled and guided and inspected. And its social effects should be monitored as well, because there would be a need. Human has a lot of desires. So if we like to start editing, then people have a lot of desires to have different characteristics for their children. So I think we have to really monitor it very closely and thoroughly. The second use is for the commercial purposes, I should say. And this is for business, as this need is in the society and people would like to make money out of this business. to promote that we can give you HIV resistance, as this Chinese guy did. He created those individuals with HIV resistance. Next slide, please. So what is the current status? When it comes to the heritable genome editing, in USA, to my knowledge, it is illegal to transfer modified embryos, and it is also illegal to export to another country where they could be transferred. In Canada, gene editing is completely banned. Even the research cannot be conducted. Some say that this is too far on the negative end of the equation, and there should be some research so that we learn and we can apply these new technologies. In UK, it is illegal to transfer genetically edited embryos, but previously the mitochondrial genomes have been transferred, but through other ways. In China, the transfer of modified embryos is not allowed. So there are some other safer alternatives for these technologies. For example, PGD, non-invasive prenatal testing, use of donor eggs, which are disease-free, donor sperm, donated embryos, and informal parenting. If the couple like to parent some of the children, then they can have those children. In my opinion, and what I read from the literature, the public is easily convinced for use of the somatic cell genome editing if it's declared safe, and the guarantee is that there would be no unwanted effects, and of course the cost is low. There is a statement from the International Summit on Human Genome Editing, and the first meeting held in 2015 and then subsequently in 2018. The statement was that it would be irresponsible to proceed with clinical use of germline editing until the safety and efficacy issues have been solved. So there is a lot of work for the scientists to really prove that this editing, what we are doing, is safe for human, because it is a matter of our future generation. Thank you very much. That was my last slide.
Thank you so much. Our next speaker is Dr. Albertini.
Thank you, Nabil, and thank you to everyone for having a chance to delve into, really a revolutionary event, I believe, in the life sciences, and it is already directly impacting our own field of assisted reproductive technology. And in fact, it's fair to say that we would not be having this conversation if we didn't do the kind of work that we did. Could I have the first slide, please? And we can, the next slide, there we go, thank you. Thank you. So, given that we're talking about gene editing in the human, I thought it would be important to just start out briefly considering what we know about the efficiency of human reproduction. And, it really started back in the 1940s with the classic egg hunt studies of Hertig and Rock, where they recognized that embryonic wastage in a sampling of over 60 women of proven fertility approached 70%. So almost seven out of every 10 conceptions that they were able to document did not result in a term pregnancy. And these were healthy people from the 30s and 40s who had families. That data and much more recent data has been considered in the study by Wilcox, which was published last year, where, again, we're not talking about embryos produced by ARTs, by IVF. We're talking about data where successful conception has been documented and pregnancy outcomes have been followed. This is a multi-international analysis. And here too, the same message came back. When you look at us relative to other mammalian species, we are not very efficient at reproducing. So what does that have to do with today's conversation? If I could have the next slide, please. The stage has been set with many of the advances that we have already heard about today for correcting inherited monogenic conditions. Many of the leaders in this field, and especially people like Alan Handyside, who were really foundational contributors to the field of PGD. Alan was a student of Bob Edwards, as many of you know, that it is well known that our patients can have their embryos evaluated for the carrier status of single gene defects. And at this point, this has become a powerful selection strategy for couples who are known to be carriers of monogenic disorders. Could I have the next slide, please? So that we're able to detect disorders, genetic disorders, that may be amenable to correction using the technology that we've heard of, CRISPR Cas9, has already given us a chance to eliminate from a pool of embryos for any given couple the passage of embryos that would contribute to inheritance of a disease state. And these two articles, which just appeared in Nature Communications, I strongly suggest for those of you interested in delving a little deeper into the nature of the problem that CRISPR and other things are now causing, pull together some of the facts from the basic research that's been conducted on the CRISPR-Cas9 gene editing procedure, and where we will see this proceed in the future, especially with regard to the ethical considerations that we will be discussing today. And I would just like to note that this is not a matter of when it will be done. This technology is already being used widely in biomedical research to manipulate the genetic structure of cells and culture, but it has also already been used to alter inheritance patterns in a variety of animals. There was very recently an amazing paper that came out of Australia on using gene editing to modify a marsupial. And it was done using much of the technology that we're going to now take a closer look at. Could we have the next slide? We're on a very slippery slope with respect to the proposed uses of this technology. This article that appeared in Atlantic Magazine earlier this year has already shown the power of our PGD technology and other diagnostic procedures, because some countries, in this case, Denmark, have within their social health care system the opportunity for all couples to have those embryos screened and given the patient's choice as to the continuation of the pregnancy. It's predicted that the incidence of Down syndrome in this country will be virtually eliminated within the next 10 years. Just something to really keep in mind as we move more deeply into the implications of this technology. Next slide, please. So CRISPR Cas9 and beyond. These were the questions posed by Nabil, and I'd just like to add a few comments before we move on to our discussion. So what is the potential of CRISPR in IVF? As was suggested, could it be used to repair single gene defect mutations, prevent the pervasive aneuploidy that we know exist in human embryos, or could it allow for modifications in the genome of the embryo that might enhance its ability to implant, to improve the incidence of implantation? Well, from curing a monogenic disease and eliminating this for a given lineage of individuals who might be subjected to this technology, also comes the power to enhance fitness, mental fitness, physical fitness. We already know that things like disease resistance, which were at the heart of Dr. Hu's experiments that caused so much concern, disease resistance using CRISPR-Cas9 is widely used in the plant and animal genetics field when it comes to agricultural purposes. So we have gained a measure of control and stand at the doorstep of deciding whether or not we should use this to modify our species. Can CRISPR solve other IVF limitations? And certainly there are many of them. Like, it's been recognized for years that of the many oocytes that we're able to retrieve from patients after controlled ovarian stimulation, a relatively small percentage of those that are fertilized are able to make a baby. And now that we are seeing more and more utilization of single embryo transfer, this data becomes much more powerful in terms of what it's telling us about the human condition. Certainly, it's telling us that this is not simply about the maternal age effect and genetics, but the many other factors that go into our assisted reproductive technologies. What about the ethical considerations? To me, we're just getting started with a conversation that is long overdue. Could I have the next slide, please? Let's just take one very quick perspective of the technology as we know it today. As was noted before in the one experiment that we've heard reported from China, the CRISPR-Cas9 complex was introduced at the time of fertilization. But our understanding of the biology of what happens to the human genomes at this critical point in time has really been expanded recently. And we've come to understand that the one-cell zygote, the zygote that you recognize at the pronuclear stage, is the first time when these genomes, maternal and paternal, are becoming significantly reorganized. And in many ways, when you consider what happens to the sperm head, it's a matter of reconstructing the chromatin in the male pronucleus. It turns out that for targeting CRISPR-Cas9, the work of Metalipov, among very few others, has shown that this is an optimal time for targeted deletion or insertion, because the chromatin of the pronuclei is in an open configuration. That said, this is an optimal time to expect off-targeting, depending on the sequence matching and availability of this unfolded DNA. There's another biological point that we must keep in mind, because any substitutions, whether they be insertions or deletions that would be implemented at this stage of development, one would assume that the first cell cycle involving DNA replication post-editing would be complete and inefficient if it were to be able to be passed on to all of the daughter cells in the embryo, both those giving rise to the placenta and those giving rise to the inner cell mass and eventually extra embryonic membranes in the embryo itself. What we know is that this seems very unlikely because implementations in the DNA structure cause temporary delays in both the replicative phase as well as the divisional mitotic phase of the cell cycle. And as Metalopov himself has shown in both rhesus monkey embryos and in human embryos, the editing is not transmitted faithfully to all blastomeres. So this is a very important point to keep in mind. Could I have the next slide? Okay, so where do we stand? The prospect is very real. The how, when, and where is unclear, but it's certainly deserving of the very serious conversation that really has yet to begin. We are in the midst of a scientific revolution, and it's important to note that in countries like ours, here in the United States, we have reached an all-time low in science education. Our science illiteracy quotient, which has been known to be low for 25 years, has now reached, we're something like 15th on the scale of developed countries around the world. So bear that in mind in terms of our audience, if you will. We also know that in the past few years, the measure of misinformation and distrust have combined to create a perfect storm for misuse and exploitation of technologies, sowing the seeds of eugenics again. Could I have the next slide? So here are just a few of the ethical considerations we may want to take up in our discussion. You know, in May of this year, the International Society for Stem Cell Research issued its guidelines on genome editing. And the bioethics community has really taken charge to the problems associated with the scientific community's perception of how we should proceed. There's been a lot of dissension and disagreement over that report. Certainly the time is overdue for having this conversation, and I thank Nabil and Progenesis for creating this opportunity. And in many ways, for those of us who worked on the cloning of embryos, nuclear transplantation back in the 80s and 90s, we have seen since that time a growing mistrust towards the scientific community, too much power in our hands. And it is time to admit that our field, assisted reproductive technologies, have really re-inspired the conversation of the eugenics movement that started back in the 1930s, a matter of concern for us as we look to communicating the truth in the future. I believe the next slide is my last slide, that we need to keep in mind that this revolution that we're in has followed down a path that consistently draws upon our field of reproductive medicine. And it's up to us to not only ask what's next, but to take an active role in seeing that this technology is used well. Thank you very much.
Thank you so much, Dr. Albertini. And now we're going to open the discussion. So I'll start with you, Dr. Sharara. So what is your read to what we have discussed so far?
I think CRISPR is probably the most significant discovery since the double helix in 1953. This is an awesome power, but at the same time, it's extremely terrifying. When we're talking about genome editing, all of a sudden, all the fears that Dr. Albertini presented in his excellent talk, and the introduction of not just eugenics, but the haves and have-nots, the implications for society could not be bigger. People with money may elect to enhance. That's what Dr. Albertini introduced, the issue of enhancements, rather than treating real diseases like Huntington's, for example, or other things. What if this technology is available? What would prevent a couple with money to ask that their kids be basically like Michael Jordan, for example, or have blue eyes and blonde hair, and all these issues that we all have been struggling with? I agree with Dr. Albertini. We have to, as a society, we have to take a much more active role in what's going on. This reminds me of the early days of IVF, when people were going down the streets and protesting and saying this is the devil incarnate, and all these things that we're doing to interfere with nature, which we definitely are. Okay, we're helping people that should have been weeded out of the gene pool to have parents. But this is what science does. It moves forward, it does not move backwards. When we have a technology like CRISPR, Cas9 or other, there's more than 13 Cases actually out there. When we have this technology, we have to use it, but we need significant oversight. And I would recommend that we should oversight ourselves rather than waiting for governments to come in and completely ban such technologies. I would strongly recommend to anybody who's listening today, Isaacson just published a book, a couple of months ago called The Code Breaker, which is all about CRISPR and what's going on. And I highly recommend people read this book. We've all read over the years what's going on with CRISPR, but this is a book that starts from the beginning, from the gene and from the discovery of the double helix all the way to the present moment. And if you want an easy to read reference, well, easy to read for us, I have friends of mine who are not scientists and reading this book, and they're having a very hard time following through on the concepts out there. But for us, this is a very easy read, and I highly recommend everybody reads this. And again, I echo Dr. Albertini's thank you for Nabil to introduce this topic, which this discussion is way, way overdue. We're behind, and we need to take a much more active role in what this means for us moving forward, not just as a society, as fertility docs, and even as ethicists in this society.
Thank you so much. Statmore, what is your inputs?
To follow up with what Fadi said, that for the last 19 years, I'm a physician at the Jones Institute, and I had the pleasure of knowing Dr. Howard personally, and hearing about the stories about the early IVF and the meeting with the Vatican, and just being able to take care of married couples. And we need to progress, and now IVF is pretty much mundane and taken for granted. So you can look at the positive side of gene editing. I mean, and as Dr. Javed had talked about, there's so much wastage of embryos, and with PGD and PGT, we're throwing away many embryos. So there would be a huge benefit in being able to correct the embryos rather than to discard them. I agree that we have still a lot of issues as discussed with mosaicism and gene editing, and with affecting other genes that you may not want to affect. But with time, these things, and with doing it the proper way, to do it like the mitochondrial DNA replacement in England, to do it with the proper ethical boards, do it in steps, do it based on science, do it in animals first. I think we will get there. And I agree with Fadi that it's very important for us to regulate ourselves, but there's so much potential with this ability. And as you know, and we may find other enzymes that may be more exact, or the technology will improve. So I think it's a very exciting field, and eventually it will benefit us as reproductive endocrinologists, but we have to do it scientifically and do it in the right way, and do it very slowly.
Thank you so much. So one of the biggest criticism to the study that was done in China was that there was no consensus among scientific community and the public, and that the decision of going and manipulating human genome was pretty much unilateral. How that research or that clinical research should look like if we had to embark in a similar experience. And I'll start with you, Dr. Albertini.
We're really in territory now that we've never been in before. I think, as Laurel just mentioned, even the technology is so rudimentary now. There are so many variants of Cas9, the Cas enzymes. There is so little known fundamentally about achieving specific targeting I think we need some years of some good science before we even start thinking about applying this to human embryos. Yes, for monogenic disorders, this is a patient population that could certainly benefit, and I think as was just mentioned, the UK's approach to the mitochondrial transfer technology for dealing with patients that are harboring mitochondrial-based disorders is a very pragmatic one. It has gone through review of religious organizations, ethics organizations, scientific foundations, etc. I think they've done their homework, and they now have a very well-planned initiative in place before any steps are going to be taken to do this on a human embryo. These being, like the spindle transfer techniques. So I would look to the UK as a model, if you will, for steps to take at this point. That said, there is around the world an active movement to move these technologies directly into practice. Okay, as you know, in Greece now, patients with mitochondrial disorders can get treated. They can get treated with spindle transfer technology. So again, we're going to, I think we're going to face major inconsistencies because this is now a global problem, okay? And at the very least, if we want to be able to move this field forward for patients in the United States, then, as I said, I would start with the foundation that's been built in the UK and proceed from there.
Yeah, so a low limited scope of research on embryos and maybe going from there. One of the questions that have been raised or criticism that was directed to the study that was done, the first study on human embryo with CRISPR, was the safety. How do you do a safety study on something that is eventually leading to life?
Well, Nabil, my point is science is, telling the truth. If we have done a research experiment in one lab, other people must be able to prove it. So for example, if we have a lot of repetition of the same experiment in different animals and there have been no adverse effects, there is no off-target changes in the DNA, we have to prove, and we have to make sure whatever we say is 100% right. I think then the public will get a kind of confidence that yes, this knowledge works. We have to really first prove it, and then those proofs would be used to convince the scientific community as well as the general public to make sure that whatever we say is right. For example, in IVF or in ICSI, there may be in the beginning, there would be some sort of resistance to those technologies, as we have seen in the history. So similarly with this technology, we have to really make sure that this technology is really tried in other experiments. There may be, I think, a lot of other ways to prove as the other technologies have been done. It has to be experimented. Our knowledge must be complete, and then at the end, when we are 100% sure, then we can move to the change or editing of the human genome.
Thank you so much. Dr. Sharara, would you like to comment on this?
Yeah, I mean, the whole thing with Jiankui He, I think a big part of it was he wanted more about fame and recognition and more than anything else. He was dissuaded from doing this trial by many, many people. Everybody he spoke to, except one person, told him that this is crazy and he should not move forward with this. And that HIV is not a deadly disease. Millions of people are living with HIV and they're perfectly fine at this point. And to go and move straight with human trials, there was no IRB. What he submitted to the Chinese authorities was very different than what he did. Even people in China were completely surprised at what he did. In terms of the Communist Party in China, they were absolutely clueless to what he's doing. But this is a guy who's a PhD. He got other fertility doctors involved, and he had them make the injection along with the sperm of the CRISPR into this. So there was no oversight whatsoever. Okay, so jumping to something like this in humans at this point is way, way too soon. We need to have a much bigger discussion with society at large, where we are at this point, where we're going, what's the awesomeness of this technology. Off-site targeting is a huge thing at this point. We just don't know when we go and cut what else is going to be cut. It's way too soon, but the genie is out of the bottle, and we have to deal with this. I have no doubt that it's a question of when, not it's a question of if. It's just like IVF. That's it. The technology is there. We know what we need to do. We just need to do a lot of experimentation before we ever do really the first clinical trial when it has to do with this. And as Dr. Albertini said, I mean, the current scientific environment in the US is one of nearly anti-science across the board right now. You know, when you politicize science, this is what you get. You get a population that doesn't believe in science, doesn't believe you need to get vaccinated. They think, you know, these vaccines are we're introducing DNA chips or whatever into people. And unfortunately, these people are dying. So it's going to happen. It could be maybe in our lifetime. I would hope so. Maybe 5 to 10 years from now. Look where we are right now with IVF. I mean, if you came and told me 20 years ago, we're at a point where we're doing PGTAs and other technologies on the vast majority of patients who are older than 35, and we're transferring a single embryo and getting pregnant and implantation rates of 65 and 70 and 75%. So I wouldn't have believed this. But look where we are right now. So I'm optimistic for the future. This is technology that no doubt will be of use. We just need to find the right application for it.
Thank you so much. Dr. Statmore, if this technology becomes available in IVF, what would be the most important limitation or bottleneck in IVF that you think this could be useful for?
Well, I'm thinking, I know Fadi and I were in Dubai a few times together, and there are certain societies where there's a lot of consanguinity, where there's limitations to embryo freezing. So I would think that they look to start with single gene defects and start with trying to reduce delivery of a baby that's going to have some severe disease that has no lifespan. That's the least controversial approach, and you could say that there's ethics involved, but it may be ethical not to do it. I mean, you have to look at unethical not to study it and not to advance in the field. So there's ethics on both sides, and you just have to do it correctly. And I think the two biggest issues are the mosaicism. Are you going to correct all the cells within the embryo? And as Dr. Albertini talked about, too, the off-target effects and the abuse of the technology.
I mean, one thing I'd like to add is for anybody who's seen the movie Gattaca, I mean, I was watching this, I think 2006, maybe, when it came out. And I was like, this is where we're going. When the technology becomes so good that basically you're in for, society, definitely not in the US, but maybe other societies would enforce the elimination of natural intercourse to have children and have enhanced kids who will not have diabetes, high blood pressure, heart disease, all these type of things. I mean, are we going to get to this point? Clearly, these are polygenic diseases, but the science fiction part of this, now that you have CRISPR Cas9 in there, definitely it's hitting home pretty closely. And, my worry is about the slippery slope of where this technology is going to lead. And again, I keep stressing the point of haves and have-nots. Are we going to have the society of, of the 1% that will enhance themselves with higher IQ and higher things, and basically control the other 99% to a worse degree than what we are right now? I hope this will never happen, but these are issues that we absolutely have to talk about. If this technology is going to be available, it should be available to everybody. It should not be available to people who have money. And this is for me the biggest fear out there.
So your concern is taking this technology beyond just medical need and looking at other things that are not.
When we start with medical needs, for sure, but once you master a technology, okay, I mean, Dr. Javed spoke about ICSI, okay, ICSI started with severe male factor, and look where we're at right now with ICSI. It's nearly done for everybody at this point, okay, definitely when you're doing PGTA and all the other things. Once you become comfortable with the technology and you get the results that you would expect, and this technology does what it's supposed to do, and the cost start becoming very, very minimal, you're going to say, well, why shouldn't I use it to do X, Y, Z? And we're going to get to that point. I have no doubt in my mind.
Yeah, exactly. So there are other situations where genetics have not been efficient at solving certain issues. And if you look at mitochondrial mutations, for example, the fact that the disease is inherited in, a population of mitochondrial units, it is not feasible to using a standard PGTM techniques to select against the mutation in some ways. But CRISPR technology does offer that promise. Where are we with mitochondria? Do you guys have any update? The promise of CRISPR in mitochondrial diseases?
I'm only familiar with a paper that was published in Cell 2 to 3 years ago, and it came from a group at the Salk Institute, and they were developing reagents that could target the mitochondrial genome. And I must admit, I haven't really been able to follow that, but that would seem like at least the convergence of technologies that would give you a handle on, of course, a much smaller genome. And, I don't know that, I don't myself know the nature of the mutations. But I know that the intention of this group was to take a gene editing approach that would be targeted to mitochondrial DNA. And it had something to do with putting like a digitonin-like molecule on the CRISPR to facilitate its entry. But it was 2 or 3 years ago in Cell that I saw that paper. I don't know if anyone else has heard of any advances in this field.
My view is this. I know CRISPR is the tool to correct, but then again, we have to see which is the better way to solve this approach. For example, in donated oocytes, the mitochondria is there. So people have found this easy, and in Greece and other countries, people are doing it. So it means there are a lot of copies of mitochondria in single cell or in the egg. So how many copies would you be correcting if you're using CRISPR?
Yeah.
So I think people found it a very quick and easy fix. Like they will just enucleate, they will transfer the pronuclei, or they will transfer the spindle into healthy egg, and they found it more economical and more efficient. So this was what I would like to share.
What do you guys think in the next 5 or 10 years? Do you think that these could actually be used in IVF?
I don't think it's going to be like where we do, every program can do ICSI, every program can do biopsies and do PGTAs. These have to be done in very unique centers because of the ramifications of this technology. I don't know, 10, 15 years from now, if it's going to be something where every clinic would have access to something like this. I think the opportunity to do harm is so substantial at this point that this is something we really need to tread incredibly carefully. It's an awesome technology, but as I said, it's a terrifying technology, at least to me at this point. We may discover something that is even better than CRISPR Cas9 in the coming few years. But I think we have to tread unbelievably carefully for the time being.
Thank you so much. Dr. Stadmore, what do you see the near future as far as this technology?
I think it's already involved with the cancer patients with somatic cell mutations, and there are so many restrictions in terms of working on the embryo that over the next 5 years in the immediate future, it's going to stay with patients who are already affected rather than having the risk of affecting future generations and the epigenetics and everything else. So I don't see it as part of IVF in the near future. And I agree that it would be localized to certain centers, such as like uterine transplantation or things like that. And time will tell.
Yeah, I think as long as there's a government sort of moratorium on this, I don't think human experimentation is going to proceed in the speed that anyone may like, for example. Again, we just have to wait and see where this technology is going to lead us. But at this point, we just have to be incredibly careful. Society is definitely not ready for something like this at this point. As Dr. Albertini showed, what's going on in Denmark with elimination of Down syndrome. I mean, are we going to eliminate everybody who has the slightest genetic disease out there? Are we going to remove the people with some bipolar disorder or mild schizophrenia or some other things? I mean, all the artists out there may disappear, and that as a society, we're going to lose an incredible diversity of where we're at. So again, we just have to wait and see where this is going to take us. But the genie is out of the bottle, as I said. We can't put it just back in. We have to go and control it somehow.
Very good. So when we look at the number, I've seen a quick study on the number of publication, and this is more for you, Dr. Albertini. The number of publication on CRISPR has reached an incredibly high number compared to other publication. I think they mentioned about 10,000 publication worldwide. Do you see publication in fertility?
I would say no. Okay, the burst in publications is because this has really become such a powerful tool for genetically manipulating any organism. Okay, and not so much for introducing an inheritable trait or modifying an inheritable trait, although the paper I mentioned on the marsupials is quite amazing because they turn this animal from a brown animal into a white one. It's the same animal, but they just changed the hair color genes, okay? What we see now is in most research laboratories, this is the go-to technology for doing engineering of stem cells. Where I work now, it's a stem cell research institute, and CRISPR Cas9 for us is the most immediate and efficient way to generate new cell lines that are carrying, very specific markers, et cetera. But as to my knowledge anyway, and I read a lot, I haven't really seen the technology used in the context of reproductive medicine.
Yeah, just what Jian Kui he did, and never published the paper. He just presented it in Hong Kong and created all this publicity on, and he never wrote it. We don't know what's going to happen to those three girls. The government in China is watching them pretty closely. We just don't know. We have no follow-up on whether they're growing normally or not. What's going on? But he got a 3-year sentence and he's in jail.
Yeah, the interesting thing that I found in that study, though, is that the mutation that he created is new. The variation in human that gives the resistance to IVF, it's very different. It's a different type of deletion. So he created one that wasn't human have not experienced in their evolution.
But Nabil, all three girls have different expressions of this. So he didn't get 100% expression of what he wanted to do. Every girl got a completely different expression of that gene, or lack of.
Well, we are getting close to this webinar. I would like to give you guys a minute to introduce your practice or interest. And with that, we will close the webinar. And I will start with you, Dr. Albertini.
Yeah, well, I think we got the conversation off the ground. I'd like to see more of this happening, and make one final distinction. We've been talking about the hair experiment in which, an early embryo at the time of fertilization was modified. But CRISPR Cas9 is also being used in the regenerative medicine field now, somatic cell manipulations, with blood disease disorders, et cetera. And there are a number of trials coming along. And the distinction that the ethicists make is that when you talk about CRISPR Cas9 for somatic cell gene editing, this is very different from the point that we've been discussing tonight, to put it permanently into an embryo. And who knows, maybe some somatic cell manipulations in the future will come to bear on the infertility field. But thank you again for having me participate. And if anyone would like to get involved with JARG, just let me know. Thank you.
Thank you so much. Dr. Javed?
Nabil, thank you very much for giving me this opportunity. I am an embryologist, and I work at the Original Fertility Clinic in Ottawa, and my interest is to help the patients, whoever need our services. Thank you.
Awesome. Dr. Sharara?
Yeah, I'm Fadi Sharara. I'm the medical director at Virginia Center for Reproductive Medicine in the Washington, D.C. area, and a clinical professor at George Washington University. One thing I'd like to add to Dr. Albertini is somatic cell, like modifications, does not have the same scariness as when we play with DNA. That's when really people think that we are playing God at this point. This DNA has been around for millions of years, and we're starting to figure out what's in it, to go and start modifying it for whatever purposes are going to be out there. It's scary. That's all I can say.
Thank you so much. Dr. Stabmore?
Yes, Laura Statmauer, and I am with the IVF Center in Orlando, Florida, after 19 years as director of the Jones Institute. And the important philosophy is to practice ART in an evidence-based way, and not be tempted by the commercialization of IVF, and to be fair to our patients and follow the science. And that's what I'm going to continue to do in private practice. And I learned that from the many years in academic medicine.
Thank you so much. Thanks to everyone for this wonderful discussion on CRISPR technology and in reproductive medicine. In 2 weeks, we will have another topic on a different subject, and I will send some notification to let you guys know. Thank you so much, and I will see you soon. Thanks to everyone.
Thank you.
Bye, everyone. Thank you, Nabil, for a great presentation.
Thank you so much. Thank you.
Bye.
Bye.